Reading up on the history of the Oxford Botanical Garden for this meeting, I became interested in Jacob Boppard, whom Simon mentioned in his introductory talk. And all the sites I found said that Boppard was born in Brunswick in Germany and was a mercenary before he was hired at Oxford. Now, knowing Brunswick well, I was curious and searched some more, and I found a nice paper on Boppard, it's cited here by Karin Sieber 2013, which makes clear that Boppard was actually born in Gdansk in the Kingdom of Poland. One finds his name easily among Gdansk aristocratic families and belonged to a wealthy family of traders. He studied medicinal plants long before coming to Oxford. And this is why he is here depicted in this famous engraving from 1713 with his Aesculapian staff. And you have all seen this gate as the background of those staff and speakers from Oxford. The stork above Boppard is the symbol of Gdansk. And as Simon mentioned, Boppard was permitted to plant vegetables to supplement his salary. And so with this staff, he is keeping this plant-eating goat at bay, doing this so successfully that the dog is not needed and is watching sleepily. This little tidbit about Boppard defending his crops from an herbivore brings me to my topic: symbioses between ants and plants that match human farming systems. I have to actually get off okay, that match human farming systems. Modern work on this topic actually started at Oxford. More precisely, it started with this benchmark paper by Camilla Huxley from the famous Huxley family. She published this seminal paper on observations and experiments that she carried out in Papua New Guinea. And when my then new graduate student Guillaume Chomicki and I were thinking about ant-plant interactions, and more precisely the evolution of ant-plant interactions as the topic for his dissertation, Guillaume visited Camilla and was hugely inspired by talking to her. Now, in her key experiment, Camilla used radioisotope labeled sulfate and phosphor that ants were permitted to feed on to prove that the plants are able to take up minerals and molecules from the feces that the ants are depositing inside the domation. This means that there must be structures analogous to roots that enable the plant to take up these substances. Now, these genera, these ant plant groups, all belong to a clade of Rubiaceae that comprises about 100 species and is confined to Australasia. So, here you see the distribution of these plants, and we decided to focus on a group of ant plants that is in the genus Cormellaria that is completely restricted to Fiji. So we got funding, and Guillaume has been doing fieldwork in Fiji every year since 2014, except for the 2 COVID years. Here you see him hanging in one of these trees, studying these ant plants, all of which are epiphytes. We obtained a well-sampled phylogeny based on herbarium material to make sure that our focal group, here circled in red, really evolved on Fiji and doesn't consist of occasional or more recent independent introductions. It really is a clade. It soon became clear that several species can coexist on single trees. Here you see 3 species of this genus, and you see on the right the phylogeny, the relationships, and you also see with their names in green new species that Guillaume discovered. We discovered several new species. And so interestingly, amazingly actually, the ants will visit and patrol different domatia, even of different species, all on that tree. The particular species of ants is unusual in having a queen live in one domation, and all the other domatia are controlled by her. These other domatia only contain workers and brood. And as the colony grows, they will occupy more domatia on that tree. Now, the first question, of course, is how do these plants ever get up there? Well, it turned out that the ant species that lives in them has the habit of taking seeds from young fruits and planting them in cracks in the bark of certain species of trees, barks or whole. Only particular species of tree have the suitable bark. Here you see one ant of this species, it's called Phyllides nagasau, carrying a seed, about to plant it, to really insert it into a crack in the bark. And in the inside, on the left, you see a seedling of our focal group on the right and a seedling of a related ant plant on the left. And marked with a blue bar are those parts of the seedling that will become the domation. And you can see that in our focal group, the domation develops above an elongated part. It's the hypocotyl. It's a particular elongated part below what will become the domation. So this helps these little seedlings to emerge from the crack, to really be pushed outside, because otherwise, if the seed is stuck in the tree bark there, it can never start putting its cotyledons out and starting its life. So if these ants are confronted with, let's say, a grain of rice used as a control here, they won't pick it up. And if they are confronted with seeds of another species that they are not, of an ant plant, that they are not interested in, they won't pick it up either. They only take the seeds of their preferred species and plant only those. We did the same kind of experiment as Camilla Huxley did. That is to say, we used radioactively labeled nitrogen and phosphor to check which tissues of the plant would be taken up, the substances deposited via the feces or pee of the ants. So we let these ants feed on these substances and then check the tissues after 10 days, both of mature plants, such as the one that you see in the center of this photo, and of the seedlings, such as the one that you see in the inset. And what we found that is the most interesting result is that the seedlings, the control is, a non-ant visited ant plant tissue, that the seedlings contained high amounts of radioactively labeled, in this case, nitrogen. You see it? So this means that the ants go in there, defecate or pee, who knows, and thereby fertilize this seedling, which is much too small for any brood or group of ant workers to live in there permanently. So it's really the case that these ants go in there, fertilize the seedling, go out again, walk to the next seedling, and so on. So now we wanted to know the details of the inner structure of the domatia of the various species. And traditionally you would have collected these and cut them open, but this doesn't give you the three-dimensionality, really the details of what's happening inside. You need high-resolution computed tomography. Max Weigand in his talk already alluded to the power of computed tomography, but the machine we had to use, of course, was a large one, because these domatia are actually football sized. And then you need the false color, you use false colors to see the different structures inside the domatia. So let's look at one of these domatia. And this particular first one that we look at is from a species that has apical growth, that will elongate and form new chambers or become larger only in one direction. And so what you see in this image is the first part colored here in red. That is what used to be the seedling, the first chamber. And then the second chamber here colored in yellow is much larger. And then there will be third and fourth chambers, as I'm showing you here, and I'm trying to get this movie. Okay, so the part in red used to be the seedling. The first chamber in yellow, the second chamber in blue, the third chamber in purple, and the fifth in green. And this is as the plant elongates over the years, it will form these chambers, and they are all interconnected. So how about the nitrogen uptake inside these chambers? We look in detail, we see that the wall of the seedling part, these walls have a warty surface. There are regularly spaced warts that you see there in the inset. And then the second chamber and third and fourth and fifth chamber, built over several years, have the smooth walls, as you see here in the inset on the right. And the ants will use these smooth walled chambers only for their brood, and this warty walled chambers only for defecation, and I want to say peeing. So the radioactively labeled nitrogen is almost exclusively taken up by the warty walled chambers, whereas the smooth chambers contain hardly any nitrogen, or take up hardly any nitrogen. Of course, the ants themselves are full of radioactively labeled nitrogen because they have been feeding on this radioactively labeled sugar solution. Here's the seedling. The seedling has a lot of the radioactively labeled nitrogen, whereas the other tissues have much less. Here you see a more detailed picture of another species of this group. And here you can see these warts, regularly spaced walls. And so we hypothesized that these would be analogous to roots. And so we used a transcriptome approach to see whether genes having to do, let's say, with nitrogen uptake might be overexpressed in these tissues as compared to other tissues. And that indeed turned out to be the case. You see the sum of the differentially expressed genes in a transcriptome of one of the species I showed you. And of course, these are only the 100 genes or so that that are relatively well known from their function in Arabidopsis. And so the 10 genes overexpressed in warty walled tissue, have to do with root development, including cell elongation and auxin responses. 2 other genes that are also overexpressed in warty walled tissue have to do with nitrogen uptake and transport. And then many genes having to do with stress responses, abiotic and biotic stress responses, are also overexpressed in those tissues. And this makes perfect sense because these are cavities filled with debris, with feces deposited there by the ants. And so, we expect that there would be pathogens, fungi, germinating in these tissues, and it makes perfect sense that stress response genes should be overexpressed there. Conversely, in the smooth-walled tissues, what is overexpressed are genes having to do with suberin biosynthesis and Rex biosynthesis, and again, this makes a lot of sense. Now, what about the reward? You would think that ant plants that regularly interact with ants have floral nectaries or extra floral nectaries, but this is not the case at all in these species that we have been studying on Fiji. Instead, where you see in the insert here on the right, you see a young bud and you see an old flower. Actually, the white petals are about to fall off, and there is no floral nectary and no extra floral nectary. But what happens is that nectar accumulates in the cupules of old flowers once the petals have fallen off. And the ants have the habit of biting into these old cupules filled with nectar to obtain the nectar, to gain access to this nectar. In this paper that is cited here, we have compared the anatomy of the nectaries of the focal species and closest relatives to really get at how these nectaries develop and how the nectar accumulates. But here I will just focus on one aspect, the exclusivity, the exclusiveness of this reward. So the co-evolved ant, Phyllides nagassau, as I said, it has the habit of biting into old cupules to get at the nectar. Now, if you test how other ants that occur on ant plants on that same tree will react to these spots, they don't react at all. You see in the first graph here, the other ant species, living in the same environment, they do not bite into the old cupules to get at nectar. And if you prick these cupules so that the nectar, you damage them so that the nectar, comes out a little bit, then the other ants are perfectly happy to take it. And very interesting is this experiment. If you place a drop of sugar onto one of these cupules with a syringe, then the other ants, again, perfectly happy to take it, but our focal ant is a bit confused and doesn't really, is not used to having its nectar sit there on top of the flower. This is an extremely unusual reward system. So now, do all the species that may coexist, or ant plants that may coexist on such a tree, follow the same strategy? Let's look at inside a domatium of one of these coexisting species with diffuse growth. In this case, the domatium will enlarge and form new cavities sort of around the domatium. Of course, not here where it is attached to the tree, but it could be formed, grow here or here, okay? Let's look inside. And what we found with our computer tomography and false color imaging is in these species, in species that have domatia with diffuse growth, there are unconnected chambers, each with its own entrance hole from the outside. So you have, for example, the purple chambers here, cavities, okay? And then the green cavities and the red cavities, but they are not interconnected. The ants living in the purple cavities cannot visit the ants living in the green cavity, okay? And as the plant ages, as the domatium ages, you see this in the inside and the upper right, this becomes really intricate. Here are maybe 12 independent apartments, each only being accessible from the outside, but not being connected inside. So this then raised the question of the holes. We have a whole study of these holes. So in our focal species, the holes in all these ant plants, the holes in the domatia are entirely formed by the plant. They are not cut by the ants, as you might have thought. And so in the focal species that are occupied by the specialized ant Phyllides nagassau, you can see that the holes have a very particular size that exactly fits, in this case, the head diameter of the specialized ant. But in the other ant plant species that coexist on the same tree, as the plant ages, the domatia holes, the holes in the domatia will become irregular and larger, and so other ant plants, other ant species may enter, and even other organisms such as cockroaches and small lizards and other organisms. As the plant ages, these holes become larger. But in the Squamillaria species, the holes stay the same size throughout the life of this domatium, which can be several years. So going into the details of the inner wall structure of those species that have the diffuse growth, they also have diffuse wall structure. So you have a little bit smooth walls and somewhat warty parts of the wall, but it's intermixed. And there will be ant brood and antification throughout this cavity. And not surprisingly, the nitrogen uptake also is less differentiated than in the first species I showed you. So here, the smooth-like wall part has taken up nitrogen, but the warty-like wall part has also taken up nitrogen. It's less differentiated. So this raised the question of who obtains more nitrogen. Is it the species occupied exclusively by the specialized ant? Or is it a species occupied by ever-changing species of ants? To answer this question, Guillaume carried out several 16-month-long exclusion experiments in which he kept the ants from visiting Domatia by using tanglefoot, sticky substance applied around the trunk of the tree so that ants could not reach the Domatium they would normally have visited. And here you have a species on the left that is occupied by various ant species. And here's the radioactively labeled nitrogen it has taken up over the 16 months. And here's another Domatium in the same tree from which the ants were excluded. And of course, it has less nitrogen, but the difference is not that striking. But in the specialized species, 2 of which were studied here, that is normally occupied by Phyllides nagasau, over 16 months, the occupied Domatium has twice as much nitrogen than the ones from which ants were excluded. This whole thing is context dependent, as any biologist would have expected. And there is a whole set of experiments reported on in this paper. Let me just give you the upshot. So Domatia planted, the seeds of which were planted by the ants in full sun. So the plants developed in full sun. Of course, they have tons of flowers, and each floral cupule will have this specialized reward exclusively accessible for Phyllides nagasau that bites into the cupules to get that nectar. So great. In the full sun, the ants get a carbohydrate-rich diet. They also petrol these plants much more than they would in the shade. So these plants have almost no herbivore damage. Conversely, in the shade, the ants don't get that much nectar. They don't walk there. They don't want to petrol these plants. And so it's much more herbivore damage. But the most unexpected part for us was what happens with the nitrogen. The nitrogen. So the plants growing in the shade actually get much more nitrogen from the ants. They're all occupied by this single species of ants, right? Because in the shade, the ants have to capture insects to feed on them, yeah? Whereas in the sun, they get all this wonderful sugar, so they don't feed on insects. So the plant gets much less nitrogen when it's cultivated in full sun. Not surprisingly, the ants act in their own interest and plant the seeds mostly in full sun, but for the plant, it gets more nitrogen in the shade. Now, what about those Domatia that have different species of ants living inside them over time and also over space? So here you have one of those. It has one species of ants in this Domatium here, and these ants will enter through the holes from the outside. They will never walk over to this apartment here, occupied by another species of ants. And maybe this apartment is empty. So here's the turnover in ant occupation in some of these species. No, in all these, actually these are all the species in the genus Squamillaria. So the 3 species that are occupied by various species of ants over time. Guillaume has observed this now since 2014. And so in some years, maybe it is occupied by that species, and another Domatium next by, or in another year, is occupied by another species. But then there are cavities that are empty, cavities that will not receive any nitrogen because there are no ants in there, so no nidification, no fertilization. Whereas in the specialized system, you basically have an occupancy rate of 99.9%. This raises many interesting questions about the coexistence of mutualists. And we have used the empirical observations on the occupancy rate and the species of ants that live in these Domatia, gotten together with a mathematician to develop a model. The empirical data also include interactions among these ants, because as you can imagine, there are larger ant species that are more aggressive and smaller ant species that are less aggressive. And so Guillaume tested the aggressiveness, the ability of these ants to coexist on sugar solution that he placed nearby. He also introduced brood of one species into the nest of the other species to quantify the aggressiveness of these, and the coexistence of these mutualists, and so nearby, living so nearby and walking on the surface of one and the same Domatium is just an amazing thing. in terms of study questions. So how old is this system? The plants, again, this phylogeny comprises all the species on Fiji in this clade. The plants are about 3 to 4 million years old. And the ant species Nagazao is between, let's say, 6.5 and 3 million years old. And it is distinguished from all the related species in this genus by having lost the ability to make curtain nests. All the other phylogeny species build their own nest in the trees, but this one cannot do that anymore. It is entirely dependent on the plants, on the whose seeds it plants, whose seeds it fertilizes. So it's growing its own nests, future nests, but also depends on these future nests. So I think I have illustrated that this ant-plant interaction is analogous to human farming systems. We have the habitual planting, we have cultivation, precision fertilization, and defense. We have the harvest of an exclusive reward, and we have mutual dependence. So is this sustainable? Is this a sustainable farming system? In the years that we have studied this on Fiji, the coexistence of these specialized, only occupied by Phyllodes Nagazao ants that are entirely dependent on this domation for their nesting space, and of the others that are occupied by varying species of ants, appears to be stable. But what will happen under climate change? The specialized system, as I said, is always occupied like 99.9%. Phyllodes ants are happiest in warm, lowland and intermediate altitudes. And diversity and abundance is generally highest at lower altitudes and in warm climates. But the diversity is reduced the higher up in the mountains and the cooler the climate. In fact, we have also studied the breakdown of these mutualisms. These mutualisms break down relatively easily. This one, we have not found a breakdown yet. And it may be that with climate warming, this completely stable interaction between Phyllodes Nagazao and its preferred plants that it cultivates and fertilizes and defends might actually benefit compared to the other ones that depend on a diversity and abundance, great abundance of different species of ants. But this needs much more study, fieldwork really. So I want to end with a picture of my wonderful collaborator, Guillaume Chomiki, who is now a NERC research fellow in the School of Biosciences and the Diversity of Sheffield. And I also want to end with a few sentences about why this is important. These ant-plant interactions are, of course, a subset of, or an example of mutualisms. Mutualisms are a huge topic in evolutionary biology and conservation. In evolutionary biology, because of the super interesting question of why selection doesn't, which normally favors selfishness, how is it possible that these mutualisms have evolved and persist? And in terms of the conservation of interactions, in fact, Pete Hollingsworth also alluded to this very briefly. Studying the genomics of these interactions, which is only possible now, is a huge topic. So I want to make a plug here for ant-plant interactions are part of the study of mutualisms, which is just super interesting and important. And with this, I want to thank you and stop sharing my screen.